We're going to cover today a lot more detail replication. What is happening during replication? First thing that happens, DNA has to separate. You have to get 2 strands of the parental DNA, and then you are going to use each one of these strands to make the new DNA. So here we are going to synthesize new DNA on this way, new DNA on that way. You are going to get your brand new DNA. One of the strands is going to be from the parent cell, and the other one is going to be the newly synthesized DNA. That's the main idea of how the DNA gets replicated. Now, this we covered last time. What are the differences between the eukaryotes and the prokaryotes replication? Here, keep in mind that you have, in the prokaryotes, you have a circular DNA structure. And remember, most of the time, this structure is supercoiled. And also, when you start to replicate, it starts at a specific point, which is called the origin of replication, the ORC. Now, one idea that is a bit difficult to keep in mind is, what is ORIC? Okay, what is this origin of replication? It's not just a name that showed up in a piece of DNA. It is actually a sequence in that bacterial DNA. It is a specific sequence. It has a name. Not only a name, ORC, but it has a TTA, TTC, CTT, something like that name. So it is a piece of DNA, a physical piece of DNA, and some proteins are going to go there and recognize this sequence. It's going to interact with that DNA. And in the bacteria, you have only one spot in the whole circular DNA that has this specific sequence. You don't have 2, 3, and 4, only one spot. Then you have your proteins that are going to get in there, interact with this sequence in a specific manner, and start to unwind this DNA and start it to replicate. So first thing that happens here, you're going to have this sequence of DNA here that is going to be recognized with some type of protein. Those are initiator proteins. And anybody remember what they're called? No, the histones are what makes up the nucleosome interacting directly with the DNA. Okay. Also, before the DNA replicates, it's going to have to get a little bit unstuck from those histones, because the histones are going to keep it very compacted. Nothing can really reach the DNA if it is really stuck to the histones. Those are some helicases. Yes, they are going to start to unwind. These are initiator proteins, and they are what? DNA A proteins in prokaryotes. So here we have, first goes for the DNA A, that will recruit DNA B and C. So here goes the DNA A up there, initiator protein. They're going to bind to this sequence of DNA, this green sequence here, and they're going to start to interact with it and wind it up a little bit. Now, if you wind it up a little on this side and you add a positive supercoiling to this side, you will have to add some negative supercoiling to another side. That means it's going to open a little bubble here that is going to allow for these 2 strands to be separated. So now when these 2 strands are separated, what happens? Well, first thing you have to remember is that naked DNA is in danger over there. You cannot have naked DNA. It's going to be attacked and it's going to be killed. This very totally unfriendly cellular world for naked DNA. So the first thing the DNA does is to associate with things that are going to shield it. So it's going to associate with single-strand binding proteins, these little purple things here. They're going to bind to the single strand and stabilize it and protect it from nucleases, from endonucleases, from anything that's going to attack the DNA. Single-stranded DNA, no good. You have to have interacting proteins. Let's take a look here at this little movie, just a little bit of it, so whenever you have the time and you go over this lecture, you can go back and watch it with more time. These are just the movies that are directly connected to the book site. Okay, so those are your interacting proteins. They recognize the green piece, they interact with it, they bend it, they start to coil it up, coiling, it's going to open somewhere else. Now, that naked DNA is going to be protected, is going to be shielded. And you get other helicases there too. They're going to keep on opening it up. You have gyrases those are the proteins here protecting the DNA. You have gyrases that come at the end to help to keep unwinding the DNA. This thing has to replicate to both sides. And if you keep on opening on this side, the other sides are going to get coiled and coiled up and coiled up. So you need to have these gyrases here. They are going to relieve the stress. Then at the replication fork, we're going to see more at this lecture. Okay, now coming to the replication fork. We know that here at the theta replication, as I said, you have to unwind all the supercoiling here. You have the gyrases that will relieve. So when you look at the fork, you know that this replication is going to go in this direction and in the other direction. It has to be bidirectional. This is very important. It doesn't happen only in one side. It has to go through the two sides. You are going to end up here, if you imagine two rubber bands, you're going to pull them apart, you're going to end up separating them both. So in order to do that, you have to have this enzyme that is going to work on both ways. Now the first thing you think about enzymes, first thing that should pop up in your head is that first thing, what is very important about enzymes is its shape. So it interacts with specific things, and it will only interact with whatever is meant to interact with. What that means is that if this polymerase is polymerizing on 5 to 3 direction, how is it reading the DNA, first of all? It is going to synthesize 5' to 3', but it's going to read this DNA strand on the 3' to 5' direction. Okay, it's going to synthesize 5' to 3', but it's going to read on a 3 to 5' direction. So this strand here that is going from 3' to 5', that's going to be your leading strand. It's a continuous replication. Now the other one here, that's where you have the problem, because this enzyme, the DNA polymerase, cannot synthesize backwards. It can only synthesize on a 5' to 3' direction. So now the cell has to have a very ingenious method of solving that problem. But then before we get to the problem, we have to remember that the polymerase cannot synthesize DNA out of nowhere. In order to start synthesizing here at the replication fork, it needs a free 3' OH. How is it going to get the free 3' OH if this is the opening of the fork, there is nothing in the middle? So in order for it to start synthesizing, it needs a primer. It needs something that is going to get in there and make this free 3' OH, so DNA polymerase can take over. And what makes that is Rna Primase. So this primase gets in there, makes this little sequence of RNA. It's not DNA, this is RNA. And this is a very small sequence. It's about 5 to 10 nucleotides, very, very tiny. But now you have a free OH over there. So now the DNA polymerase can go in there. This is DNA polymerase 3. It will recognize this free 3' site, and then it will start to synthesize.
C is for clue, that's okay with me.
This is very important. No primers, no DNA replication. You need to have the primer. This is a site that is nice for you to have an idea of this supercoiling, the second dimension, the 3-dimension DNA. When you start wrapping up these structures and get it compacted, you can watch this also when you have the time. The histones and the sequencing, how you pack up the DNA. So I put it there for your enjoyment and memorization. Now, this is a slide we had stopped last time. Right, so this is how did people find out about DNA polymerases. It happens that this scientist here, Arthur Kornberg, he discovered DNA polymerase 1. And then his son here, Thomas Kornberg, discovered DNA polymerase 3. Polymerase 1 was about 1950s, and polymerase 3 around 1970s. Not even I was born at this time. What are the properties of these polymerases? well, DNA polymerase 3 is actually more important than DNA polymerase 1 in a sense that it will synthesize the new strand of DNA. It's not polymerase 1 that synthesizes the new strand. Polymerase 1 has to do with something with the primers and remaking DNA, but not the synthesis. Synthesis, most important, DNA polymerase 3. Now, what are the properties of this? As I mentioned, it will make a new strand of DNA in the 5' to 3' direction. And here is a little illustration of how those bases will look and how they form this phosphodiester bond. And also, when you make this bond, the new nucleotide that's coming in, see, this is a nucleotide, which means it has 3 phosphates attached to this. In order to synthesize DNA, you need some form of energy. This is not just a natural exergonic reaction. This is a reaction that requires energy. Now, where is the energy coming from? It has to be something with phosphates. In this case, it's not coming from ATP alone. It's coming from ATP, GTP, CTP, TTP. What does that mean? It means that all those have 3 phosphates. Where does the energy from ATP comes from? From the phosphate groups. Okay, so here, you know, ATP is the most important form of energy we have in the cell. But when we're making new DNA, all of the 4 nucleotides are important for energy. So here, when you make this bond, you are cleaving a pyrophosphate, which means 2 phosphate groups together. When you do this reaction, you have this leaving group that is a pyrophosphate that leaves the reaction. And this is what drives the reaction. That is the energy for the reaction to happen. And also what is important to know about the polymerase is that it will make matching nucleotides. If it reads A, it's going to bring in a T. If it reads C, it's going to bring in a G. It has to make exactly the same match, perfect match of nucleotides that is complementary to your initial strand. This is very important. This polymerase is very clever. And it's so clever that when it finds something that is not a perfect match, you know, it's a human enzyme, it will make mistakes, right? Or in this case, it's a prokaryotic enzyme, polymerase 3. It will make even more mistakes. So when it finds something that is not matching, for example, if it finds something that is C connected somehow to A, it knows that this base pair is not right. So I'll have a little movie here too. It's going to back up a little bit and correct that and then keep on going. So the analogy for how this thing does the correction is when you are typing and you put a typo in there and then you use your backspace and you go back a little bit in the sentence and you just go back and correct it, put the right one and keep on going. That's the image you can keep in mind for the correction part of this RNA polymerase. Now, how is this polymerase composed? What is it composed of? What is the structure of DNA polymerase? It's actually a big molecule here. It's made up of several pieces. It has 3 subunits that make this core enzyme. So it has an alpha synthetizer. A second here. We're talking about prokaryotes. So what you have here, several subunits of this polymerase. You have an alpha subunits that makes the nucleotides, that will make the elongation of your DNA. And you also have another subunit here that is called epsilon, that is the proofreader. This is the yellow piece right here. So what happens is that after this alpha subunit puts in the nucleotide, it will be checked by the epsilon. If epsilon say everything is in order, the polymerase keeps on going. If epsilon finds a mistake on the base pairing, it will cause this DNA to back up a little bit and correct its mistake, okay, cut out the piece that is wrong, correct its mistake, and then keep on going. Now this epsilon here needs a little push, needs a little incentive to keep on working. So it gets a little push from this blue subunit on top here, which is called the theta stimulator. Okay. Now, how is it that the DNA polymerase gets stuck to the DNA and keeps on synthesizing new strands of DNA? Actually, the DNA polymerase has a very low affinity to the DNA. It will bind to the single-strand DNA to start making the new strand, but if it depends on itself, it will fall off very quickly. So it will bind and then make a little DNA, and then it will fall off, and then maybe it will find another piece of single strand, bind again, make a little more, and fall off, and then you have a huge mess, and you do not have your DNA duplicated. So in order to avoid that, the cell came up with a system that has these clamps. They're going to clamp the DNA polymerase to the DNA. Now, the idea of this clamp is that you have this donut that just grabs onto the DNA. So in order for this to happen, this is a very nice visual idea of the DNA clamp, and I believe also of the DNA clamp loader. This is a very nice little presentation. Also, when you have the time, go through it. This we're talking about the lagging strand, because when you're talking about the leading strand, once you have your initial RNA primer, the DNA polymerase is going to bind there, and it's just going to keep on going. So the clamp is going to be loaded to the DNA, is going to hold on to the polymerase, and just stay stuck together. Stay stuck forever until it finishes up that strand of DNA. Now, this is more concerning for the DNA lagging strand, because this is the part when the cell has really to come up with this ingenious configuration of how to invert this DNA in order to make 5' to 3' new DNA strand. Yes, exactly. Well, it has to get in there somehow, and when you are in the middle of the replication, how is it going to get in? I mean, this replication doesn't really work like magic. You keep in your head the idea of that ring and string trick. Watch the magician has a string and the ring, the plum, somehow the solid ring got through the string. That doesn't really happen. There is a trick to it, right? So you imagine the magician as the clamp loader. That's what he's doing. He's loading the clamp. So what happens is that when you have this clamp loader, it grabs the clamp and distorts it a little bit in order to open this donut. So now the clamp, there's going to be clamp on the DNA like this, okay? Before it clamps, the clamp loader goes in there, opens the clamp, and then this system will find the single strand DNA, and it will clamp to it. Now the clamp loader leaves the clamp in there. So now you have your DNA polymerase with the clamp. The clamp has high affinity because once it locks in there, that's the donut. DNA is not going anywhere. Okay. Now you can imagine what are the interactions between this clamp and the DNA. What do you think is happening there? How is the clamp going to be stuck to the DNA? Yes, they are positively charged. Now, because you have this circle that is positively charged, and in the middle you're passing this negatively charged DNA, where is it going to bind? It gets really confused. Okay, here you have your DNA that is negatively charged, negative charges everywhere, and now you come with your circle here, your donut, that is positively charged. But if it was only positively charged in one side, then DNA would get stuck over there. It wouldn't go anywhere, right? Now this donut here is positively charged everywhere. So what's going to happen to the DNA in the middle? It's going to float in the middle. This is one of those magnetism tricks. You have those little office devices that you have a globe in the middle, and then you have two like magnets that keep that globe in suspension. Have you seen those ones? Yeah. This is more or less an idea of what happened to the DNA in the middle of the clamp. It is attracted to both sides, to all the sides. So it just stays in the middle. So this clamp is just floating along the DNA, and then it's connected to the polymerase, and is able to keep it in place, because if it depends again on the DNA polymerase 3 alone, it's just going to fall off. It has very low processivity and very low affinity to the DNA itself. And also this clamp is what enhances the processivity of the polymerase. Okay, increases the processivity of the polymerase. Yes?
What stops it from coiling inside the donut? Because if
Oh.
you think instead of it going straight through, you would think that it would just...
Oh yeah. You can have something that the negative charge is just going to go around and do like this, right?
Why doesn't it tangle up inside?
See, the cell thought about that too. So we came up with some single binding proteins. If you have a naked DNA, remember, naked no good. So you have single binding proteins that come here and interact with that. That will keep the DNA straight. Okay, keep it in shape. Once the clamp gets close to this single binding proteins, it's going to expel them out of there. Remember, these single binding proteins have to be also positively charged, right? So they don't like this clamp coming along. Clamp comes along and these proteins all come out of the DNA, and now the DNA polymerase 3 comes right on the back of it and starts making the new DNA. Okay, but yeah, it's a very good question. Why doesn't this wrap around? Okay. Well, let's see if this loaded finally. Let's go to 2.25. So here you have at your lagging strand. Okay, now this is a very important picture to keep in mind. When we are looking at the structure of the DNA polymerase 3, remember it's made up of several units. You have the alpha, the epsilon, the theta unit. The alpha unit will polymerize. The epsilon is going to do some proofreading, and the theta is going to stimulate the epsilon to do its job. But now you know you have two strands that you need to replicate. You have your leading and your lagging strand. The leading strand, no problem, grabs, keeps on going. The lagging strand, it's a problem. You have to do it backwards. So what it does, it will flip the DNA because it cannot flip itself backwards. So something got to flip backwards here, which is that part of the DNA strand. So what happens here, this is still going to be synthesized in the 5' to 3' direction. DNA is still going to be read on the 3 to 5' direction. Okay, so what happens here? You push this DNA forward. This polymerase is going to recognize a primer as this fork is unwinding, and then you have a single strand DNA. You're still going to have your primase, your RNA primase is going to get in there and put a primer. So now the clamp loader and the clamp, those are attracted by this primer, by this RNA primer. So the clamp loader brings the clamp to that junction here. See, this is the clamp. The clamp loader will bring the clamp and the whole DNA polymerase to the end of the primer, and then it will start to make new DNA. Now look what happens when you get to the next primer. This association that only associates with a single strand DNA, loader associates with the DNA RNA primer, the clamp associates with a single strand DNA. But you know, this is a bit too much information. So just think about the clamp is loaded into that part of the RNA. Okay, clamp is loaded to RNA primer. When it gets to the next primer, that you actually have some piece of DNA in there, is going to dissociate. It's just going to come off because this part was already made. It doesn't need to keep on going forever because it's going to do the same job twice. So what happens here, it dissociates. Okay, there's an Okazaki fragment. Now it's going to recognize the next primer and is going to get polymerase there again, and it's going to start to synthesize again the new strand of DNA. Finds the other one that is newly strand, dissociates, then it goes back to the next one. And this cycle keeps on going. So please, when you have the time, sit home to study, go over the little movie. You don't have to know all the exact details, which protein, which molecule is, has more affinity to which part and which one happens first. Just to have the general idea. Clamp goes to the end of the RNA primer, then brings polymerase, you get polymerization. When it hits the next RNA that already has DNA synthesized, it will fall off because it's not needed anymore. Okay, so we talk about the alpha, epsilon, and theta. Now, as I mentioned, you have two strands to synthesize, so you need two units of this polymerase here to be stuck together. Now, what keeps this structure together is this tau subunit right here in the middle. And this is very easy to remember because if you imagine two strands of DNA here, you have to synthesize. You have this molecule that's going to grab the two sides, so it kind of looks like a T. Okay, T in the middle, grabbing on the two sides, that's the tau subunit. We'll keep all this structure here together. We'll make two functional subunits on this protein here. Okay, now the gamma loader is what I just mentioned, that is going to load this DNA polymerase into the strand to make new polymerase. That's the loader, brings the clamp. That's the gamma loader.